At the evolutionary relationships between living things and extinct organisms. And now we can take this kind of information and learning and use it in taxonomy in terms of how we classify organisms. Okay? And we said one of the things we're trying to figure out when we do systematics is we're trying to determine phylogenies. Phylogenies are the evolutionary history of a specific group of organisms that you're interested in studying. Keeping in mind that this is not something you can ever know for certain, because nobody has ever been there for the entire lifespan for that group of organisms and has watched every change that's happened over time. So all we can do is look at the data that we have, and that can be molecular data, the DNA. It can be morphological data, for example, the anatomy of the organisms, and do comparisons based on that, and come up with some sort of a hypothesis for how we think they're related. And generally speaking, the way that we represent our hypothesis, not something we can prove, and we can revise it, okay, is to use something called a phylogenetic tree, which is the branching diagram that you see right here. There are many different types of phylogenetic trees, and there's different ways of making them. We're going to focus on one main way of making those. Okay? But before we get into that specific type of phylogenetic tree we're going to discuss, let's just talk about trees in general and get a better feel for exactly how we're supposed to read these, because you're going to be reading a lot and seeing a lot of phylogenetic trees in this class, and some you're actually going to have to kind of fill in blanks on tests and quizzes, so you want to be comfortable with how to read them. So let's look at the basics first. Phylogenetic trees, remember, are drawn across a time scale. So right up here at the top represents modern day species, what exists right now. And the further down you go in the tree, the further in the past you're going. So that down here, you're at the oldest point in this particular tree. Sometimes trees can be turned to the side, works the same way. That would just mean the right side over here would be present day, and as you go towards the left, you're going further in the past. Trees are also made up of what we call roots. The root is the bottom of the tree right here, which we'll talk about what they mean in just a minute. We have the lines, all the different lines that you see here in the tree. We have the places where a branch is. I've designated those with green dots, but those green dots are not usually present in the tree. Those are nodes, and something called clades, which are going to be groups that we're going to discuss in a second. So let's talk first about the root of the tree. Okay. Going all the way down to the bottom of the tree, here's where our root is right here. This is the oldest point in time in this particular tree. Can you go further in the past? Absolutely. And with this particular tree, that's all the further in the past it's going to go. That's the oldest point. It also is going to be a common ancestor for all of the things that are represented in this tree. So you can see the lineages here that we have are mosses, ferns, conifers, flowering plants. That means that this is some sort of an ancestor for all four of those groups of plants that you see right there. Okay, is it the only ancestor? No, we're going to get to that in a second. But this is going to be a common ancestor for everything that's in the tree. Now we've got our lines, and our lines are going to represent lineages. Now what those lineages are depends on what kind of a tree you have. Okay, if you're specifically doing a tree, for example, of just the order of rodents and animals, you may actually have individual species that are on each of those lines right there. But generally, when you've got a more inclusive tree like this, where you're including a lot more organisms, a lot more species than that, you oftentimes can't branch it out into individual species. That would get way too cluttered and hard to read. So maybe your lineages then represent genera, or families, or in some cases even domains. It can be any one of those levels that we saw in that taxonomic hierarchy that we discussed in our last class. Okay? So obviously these right here are going to be more than just species, because there's a lot of different species of flowering plants. More than just families too, because there's many different families of flowering plants as well. So some higher level than that. Now keep in mind too, as you move through these, this is not just a point, this is an actual line. And we've already talked about that implied time scale here. This line represents time. So that means as you go along this line, right, the populations of the species that that line represents are changing over time. They're changing because of all of those microevolutionary mechanisms that we learned way back in Biology 130. Genetic drift, gene flow, natural selection, non-random mating, mutations, all of those things are happening on these populations as we move through time. Okay? So what the population looked like right here is going to be different than what it looks like right here. Because from here to here, some changes will happen. Are they big changes? Not necessarily, okay? But there are going to be some changes that happen because those mechanisms are always acting on populations. Okay? Now, every once in a while, you can get enough of those small changes that happen that you do get a big change that appears. And that's what these little tick marks are that you see right here in your phylogenetic tree. These represent kind of the accumulation of small scale changes that led to this new evolutionary novelty that appeared in evolutionary history. And you can tell which ones came first because there's time scale here. So vascular tissue appears first in the fossil record because it evolved first. Then you got seeds later on, right? And then you have flowers later on. So you have more plants that have vascular tissue than you do that have seeds, and you have more that have seeds than have flowers, and so on and so forth. Okay? Does it tell you exactly when it occurred? No, but it does give you a relative timeline that you see right here. So you can see that everything above this point right here, you would expect everything from this point on would have vascular tissue, with the exception of any lineages that maybe later on lost that trait, because that can happen as well. And we'll talk about some of those later on. But look for those changes in that genetic makeup and the appearance of new characters within the phylogenetic trees to kind of help you read these. And then you have your nodes. The nodes are places where you have branches occurring. You have an ancestral group right here that branched into 2 or more descendants. It doesn't have to be just 2 branches that comes off of it. Many times it is, but you can have a lot more than that. You can have an infinite number if you wanted to. But in this case, each of these only ever branched into 2 descendants. You have an ancestor, right, that kind of went in 2 different directions. You had one group that went this way and evolved into this lineage, another group that went this way and evolved into this lineage. Right? So this right here is also going to represent common ancestors. Okay? Those points represent the common ancestor for everything above them. Okay? So for example, this right here, this point, this node, this is a common ancestor for both conifers and flowering plants. This one right here is an ancestor of ferns, conifers, and flowering plants. Right? And this one down here is an ancestor of mosses, ferns, conifers, and flowering plants, because all of those branch from that point. Now, you might ask yourself and say, wait a minute, Dr. Kruger, you just told me that this point down here was the common ancestor of mosses, ferns, conifers, and flowering plants. And now you're telling me that this point is the common ancestor. The answer is they are both common ancestors. Okay? This one is just one that's further in the past. This one is the most recent, right, of all those individuals. If you go any further past this point, you no longer are including the mosses. So anything beyond this point is not going to be an ancestor of mosses. This is the last one, right? It's the most recent ancestor that we have for all of the lineages that are branching above that point. That's the difference. Everything here from here down is an ancestor then, of those 4 groups. It just goes through kind of time scale. All right. Now let's get into the very specific types of phylogenetic trees that we're going to discuss in this class. And we're going to talk about how we're going to use this information to classify organisms. And why are we using systematics nowadays in taxonomy rather than just classifying plants in any old way we want to based on common features that they have? Okay? In traditional taxonomy, you can group organisms together however you want to, with whatever common feature you want to use. Okay? So for example, we've got 2 different species of plants right here. They both have these beautiful yellowish, orange, red flowers, feature that they share. And you might say, okay, I'm going to group everything together that has that colored flower, that bicolor that we have got there right there. That's going to go together into one grouping. We're going to name it something. That's what you would do in traditional taxonomy. The problem is that these 2 species right here are not at all closely related. They're not even in the same family. This is a type of columbine. It's in the buttercup family. Okay? And this one right here, this is a type of milkweed. So it's in the milkweed family, not even closely related. And if you actually look at the anatomy of the flowers, you find that out. Right? The anatomy of the flowers is not at all similar. They look kind of like they're similar, but very, very different if you actually start dissecting and looking at it. Really, the only thing they have in common there is that coloration. That's the same between the 2 of them. But if you go into relatedness, evolutionary history, you find out that this species right here, which is sometimes called butterfly weed, Aesculus tuberosa, is more closely related to all 3 of these species right here, which are also milkweeds, than it is to this flower over here, despite the fact that all of these have very different colored flowers compared to what they have. Now, if you look closely at them, though, you'll definitely be able to see that the anatomy of the flowers are the same. Okay? They all have the same unique features on them. The shape of it, the way that the stigmatic slits are going to be is the same. All of those are similar. So that anatomy is something that an ancestral milkweed had. Therefore, all the descendant milkweeds have that. It's based on their evolutionary history. So we can group those together. because that's a feature that is based on their evolutionary history. The coloration is not, right? Because these two species right here actually evolved this coloration separate from each other, independently, at completely different times, right? And other flowers that are in this family right here are not necessarily going to be that color. So that's something to keep in mind with using traditional taxonomy. So what we're going to try and do instead is this, right? If you want to classify organisms in a way that accurately reflects their evolutionary history, use the information that you learn through systematics, right? Through systematics, we would learn that this anatomy of this flower is something that is an evolutionary trait, right? Not the color. And so by learning that information, then we can place groups of species into common units based on their evolutionary descent. Okay? So we're taking systematics, we're using that for the purposes of taxonomy, and that's where everything is going nowadays, right? Not just within plants. All things are being organized like this. Okay, so we're going to talk about a special type of phylogenetic tree that's referred to as a cladogram. Okay? So cladistics is one way of making phylogenetic trees, one way of kind of determining or making hypotheses for phylogenies. The way that it specifically does this is it uses something called shared derived characteristics, right? That can be traced back to a group's most recent common ancestor. That's what they base the whole tree on. Now we're going to get into what these are. Hopefully if you've had evolution or if you've had biology 130, you've at least scratched the surface on this. We've talked a little bit about that. We're going to get into some more detail in this class because one of the things we're going to do is as we go through the different groups of plants, I'm going to be giving you lists of characteristics that are the ones that define them, that are these shared derived traits that that group has, no other group has. And so you'll have to start being able to keep those straight, making sure you put them with the right groups of organisms and what defines them. So this concept can be kind of important for the rest of the semester. Okay, so this is how we do cladistics. Okay? You examine the character states of specific characters. We already talked about in our last class what characters are. They are some sort of inherited trait or attribute that an organism has. For example, flower color, leaf shape, things like that. Position of the ovary, all of those could be examples. And the character states were the different forms of those characters. So for flower color, you have pink, blue, white, yellow, all those different options. We talked about the leaf margins. You can have crenate, serrate, dentate, all sorts of things like that. Now, what we have to remember is what we were just talking about in phylogenetic trees, that there's an implied timeline here, right? And that as these populations go through time, they are undergoing changes, microevolutionary changes. And sometimes those can add up to big changes right here. So evolutionary processes can change characteristics over time. So if we notice where these things appear right here, and we said vascular tissue, this is a trait that appeared in evolutionary history right here, meaning that everything above this, including these ancestors right here, have that vascular tissue, unless they lost it later on. But that means that everything below here, including things that branch off down below there, don't have that feature, okay? So what that means is that we're going from one type of character state to another type of character state. The one that exists back here, the ancestors, is referred to as the ancestral character state. In this case, the ancestral character state was to not have vascular tissue, okay? But once we get to here, this is considered the derived character state, the new evolved one, right? And so that one is also sometimes referred to as an apomorphy. Apomorphy and derived character state mean the same thing. So the derived state is to have vascular tissue, right? So everything that has vascular tissue has that derived character. And you can have any number of derived characters represented in a trait. Just look for all tick marks. Like this cladogram, all tick marks represent different derived traits. You can even have them on these branches right here. They don't have to be just here. And you can have more than one in between here. You can have 7 in between here. Right? All new derived traits that appeared during evolutionary history along that lineage. Right? But you'll notice that when we talk about cladistics, you know, we're saying that it was dealing with what's called shared derived character states. This just says derived, right? What we mean by shared is that this is a trait that's going to appear in a group of animals but no other groups. It is distinctive and unique to them. In some ways it defines them. I'll use an animal example for you. Okay? Why are mammals called mammals? Because they have mammary glands, right? Mammary glands, modified sweat glands that produce milk on the females. I guess that means milk is modified sweat. We'll get into that, that's a whole other class. Okay, but that's one of their defining features. That is a shared derived trait for all mammals, is that they have mammary glands. Okay? Another shared derived trait for them is that they all have fur or hair, right? So that's distinctive just to that group. No other group of organisms have either one of those characteristics. So those are characteristics that define them. Right? Now you can see we've got lots of different shared derived traits listed in here. Right? So for example, right here, we have the appearance of vertebrae in the fossil record of evolutionary history. That means that everything above this we would expect to at least have rudimentary vertebrae, right? Or if not more defined than that. So we would say that's a defining feature for this group right here, right? We have another one right here, jaws and mineralized skeletons. That's where that appears in evolutionary history. So everything beyond that point we expect to have that, right? This type of phylogenetic tree that you see right here that includes the shared derived traits in the actual cladogram, or in the actual tree itself, and uses it to group the organisms together. We're grouping all these together because they all have vertebrae. We're grouping all these together because they all have jaws and mineralized skeletons. Right? That is specifically called a cladogram. So it is a type of phylogenetic tree that is based off of shared derived traits. And these characteristics right here define the clades, the groups. Okay, so I'm kind of waving my hand around this group right here and saying all these things have vertebrae because everything above this has vertebrae. But if you look closely here at some of the names right here, you'll see that that group is referred to as the vertebrates. They're named after their distinctive feature. Okay? Got another one down here where we've got limbs with digits. Okay? That's our tetrapods, tetra 4 pod limbs. And we've got our amniotes down here, which are going to be defined by the fact that they have an amnion. So we use these characteristics to define the groups, okay? And you'll have to do that on a test, right? If I give you a cladogram, it could be a group of organisms you've never seen before. As long as I've got the tick marks listed in there, and I circle or indicate what grouping I want you to look at, you should be able to tell me what the shared derived trait or traits for that group is, okay? All you have to do is figure out, okay, here's the grouping right here, so all of these right here, and just go down and find the tick marks that are just below that point. Okay? All right, these are important because this is what we're using now for classification of organisms. If we're talking about, just generally speaking, any phylogenetic tree, these kinds of groupings are called monophyletic groups. If we're specifically talking about a cladogram, which is a specific type of phylogenetic tree, we refer to them as clades. So those two terms are somewhat interchangeable. It's just that this one only applies to these groupings within cladograms. This can apply to any phylogenetic tree. Okay? But let's define it, what a monophyletic group is. A monophyletic group is going to include a common ancestor and all of its descendants. So you have to think back with what we were discussing as far as how to read a phylogenetic tree. Okay? What part of the tree represented common ancestors?
Nodes.
The nodes, exactly right, okay? So the nodes, the places where you had branches right here, right? Those were common ancestors. And they were the most recent common ancestors of everything that branched above them, right? So look at all the nodes in this tree, right? We, for example, have one right over here, right? And all of its descendants are everything that branched from it. So both these lines right here would be included. If we circle that node, as well as all the lines that branch from it, that would be a monophyletic group. Because the cladogram would be a clade. Right? We have another one right over here. That's you. And we've got two lines that come from that one. So if we circle that whole thing, that's another monophyletic group. Right? We have another one right here. So if you circle everything that branches from that, that would be a monophyletic group. Another one here. There's our monophyletic group for that. And even this one right here, which includes all of these, that would be a monophyletic group. You can also see that you've got smaller monophyletic groups within bigger, within bigger, within bigger. Kind of has a nested design. Oops. Smaller groupings within larger groupings within larger groupings. However, if you think about that, that's kind of handy, because that's kind of how our naming and our classification system and taxonomy works. You've got species, you've got genera, you've got families, you've got all the way up through kingdoms and divisions and domains and things like that. You've got bigger and bigger groupings. So if we can find bigger and bigger groupings, that would be a super handy way of naming things. And that's exactly what we're going to do. Okay, so you're going to take your, in systematics, when you make your phylogenetic tree like this, you're going to start looking for those clades. And you're going to start naming them. Okay? So we have, for example, a group right here. Right? This is a node right here, so there's a clade right here. If we go down in the tree and look at what shared derived traits they have, one of the biggest ones they have right there is that they have seeds. Everything from here above has seeds. So what do we call that clade? We call it seed plants. Okay? We have another one right here. This little single line is not a single species. There's a group of species out here, so that's actually a clade. If we follow that one down, there's a shared derived trait for them, so we call them angiosperms, which means flowering plants. We have another one right down here. This is a node. So we have a clade here. What is that one referred to as? The vascular plants, exactly right, because their shared derived trait is vascular tissue. And we have another one down here, and they're called the land plants, exactly right, because their shared derived trait is their ability to live on land. Okay? So that's what we're doing. We're converting cladograms into classification to identify and group and name species. And you can do this for animals, you can do this for bacteria, you can do it for anything, even plants. And so we're seeing that in all the different fields of study. Now, if you go back to this picture right here, I'm just going to use the color coordination here to help us. You'll notice that there are a couple other names here that don't have any sort of grouping like this that has a bracket around it to indicate that it's a clade. But most of us have probably heard of these before. Green algae, I guess most of us know what green algae is. We may not have heard this one unless we remember it from 130, but non-vascular plants and seedless vascular plants, my Tuesday lab better remember that one, right? Because we just spent a whole lab talking about those, right? But you'll notice if you look at the colors here, right, only the parts that are in blue right here are actually green algae. We've got an ancestor, a node here that was green algae, and some of its descendants are green algae, but not all of them. Because all of these are descendants from that node as well, and they are not blue, which means that they're not considered green algae. So why do we still have these names that exist that aren't really monophyletic groups or this new way of classifying things? Because we have some exceptions to this. There are certain types of groupings that we still have some of these old names, mainly for convenience. We'll get into that in a second. There are two main types that we're going to talk about: polyphyletic groups and paraphyletic groups. The one that I just showed you just now was a paraphyletic group. Because a paraphyletic group is going to include a common ancestor and some, but not all of the descendants. Okay? So you saw that node was colored blue because it was an algae. And some of the descendant lineages that came off of that were also blue. But there was a whole section of the tree that branched from that point that wasn't blue. And we were missing a whole group of descendants from that ancestor that are not classified or considered algae. Okay, so you can see in here, we are circling our node to include our common ancestor, but we're not catching all the branches, all the lines that come from that. So we're missing this one right here for this clade. We're missing this whole grouping right here for that one. We're missing this lineage right here for this grouping. For this one, we're actually missing this line and this line right here. Right? And over here, this one, and this one. Right? One of the best examples I can give you for this is actually the old classification of reptiles. Back when I was in high school, we won't talk about when that was. Reptiles were considered snakes, lizards, turtles, tortoises, crocodiles, things like that. That's it. Okay, so if you look at where that falls within the phylogenetic tree here for those animals, you can see here's our turtles, tortoises, here's our lizards and snakes, here's our crocodilians, right? And this ancestor down here, that's the common ancestor of all of these groups, was also considered a reptile. Right? But the problem is, we have another lineage over here that didn't used to be included in this classification, and that's the birds. They are a descendant from this reptilian ancestor right here, but they were not classified as reptiles. Right? Well, if we want to stay with this method of using evolutionary history to name organisms, we've got to include them. And so they've reclassified reptiles into what you see in the green right here. It now includes the birds. They are now considered reptiles. That makes it a monophyletic group. When we say reptile, it now means a monophyletic group. Now polyphyletic, a little bit different. Polyphyletic is made up of organisms in which their most recent common ancestor is not included. Both the monophyletic group and paraphyletic group includes a common ancestor. This one doesn't. Okay? And usually the reason why that is because whatever feature it is that you're using to group these organisms together, that ancestor didn't have it. Okay? So a good example for that would be warm-blooded animals. And I know now we use the term endothermic, but we'll use the old-fashioned term. Warm-blooded animals, things that can regulate their temperature internally through their metabolism, their physiology. If we go down here and look at this, the two groups of organisms that are warm-blooded are birds and mammals. Right? They're not right next to each other on the tree right here. Right? And you can see we're only just circling the tips up here. Because it's not until you get right here to this point in evolutionary history, or this point in time right here for the mammals, that warm-bloodedness actually appears. Everything below that was cold-blooded. And if you follow their branches down, some students have trouble with this sometimes. You want to find their common ancestor. You keep following the lines down until you actually get to the point where they would connect, okay? Where their most recent common ancestor is, and that's right down here at this point. Okay? You'll notice that the other descendants that come from that point are not warm-blooded. Okay? So we're not including them when we say warm-blooded animals. But this ancestor right here was also not warm-blooded. It was cold-blooded. So because we're not including the ancestor, we can't call this a paraphyletic group. If we had circled that and just left these lineages off, it would have been a paraphyletic group. But because we left their ancestor out, it's not. It's called a polyphyletic group. This one is usually the harder one for students to kind of grasp. Okay? One of the ways I oftentimes tell my 130 students to kind of practice this, because you have to be able to recognize these for your exams in this class. Paraphyletic, polyphyletic, and monophyletic, because I'll be talking about examples of each throughout the semester, is to just do Google search. Go online and say, examples of polyphyletic groups. Click on the images and just start looking. The more examples you see of those, the easier it becomes to kind of grasp that concept. Because you're not going to get the same examples on the test that you get here in class. You're going to get different examples. So I don't want you to memorize these cladograms. I want you to be able to recognize this concept in a different way. So practice with that, as many different cladograms as you want to make sure you feel comfortable finding those different types of groups. Now, none of these that we're using, other than the monophyletic group, are going to reflect evolutionary history, not natural. So paraphyletic, polyphyletic, usually not very useful in classified organisms because it doesn't tell us anything about their relatedness. So why use them? In most cases, we're using them still just out of convenience. We're not usually naming new ones, we're keeping old ones that already exist because changing it at this point would cause a lot of confusion. Most of us have probably heard of things like reptiles. We may not have heard of Sarcopsida. It just depends. So we want to try and avoid some of that confusion and changing things that have been traditionally used for years and years and years that people are familiar with. So we'll run across things like green algae, non-vascular plants, seedless vascular plants, things like that. But what you'll find is that when we get into those groups, they are not going to have shared derived traits. A polyphyletic and paraphyletic group cannot have a shared derived trait. Let's look at this example right here. Okay, non-vascular plants, that's these right here. Okay, they don't have a shared derived trait. You might say, well, right below them here's the ability to live on land. Yep, but that is not distinctive just to them. It is not a defining feature of this group. It is a defining feature of this group. Everything. The monophyletic group in that case that is defined by that shared derived trait is all of the land plants, right? Not just the non-vascular plants. There isn't anything that defines that group. So we'll get into that as we get into those shared or those different paraphyletic groups that we're not going to have defining features for them. Most cases, they're usually defined more by what they don't have rather than what they do. Okay, that's going to be our introduction to taxonomy and systematics. As I promised, the next thing that we're going to go into is going to be a little bit more detail about one of the activities of taxonomy, which is naming organisms. So we're going to talk about nomenclature, botanical nomenclature, to be more specific. Okay. Now, fair warning, the rules for botanical nomenclature are not gonna be the same as they are for zoological, for animals or for bacteria, for that matter, for viruses as well. They're gonna have their own set of rules. When you think about naming organisms, you typically think about two different types of names. As a matter of fact, when we got into lab this last Tuesday, we talked about how for your site recognition exams, you're gonna have to know your family, you're gonna have to know your scientific names, you're gonna have to know your common names. Technically, common names are actually useless, right? Because they're only useful in this general area. If you start going other places in the world, they're absolutely useless, because nobody understands them. There's a lot of reasons why that is. But we like them, okay, because they are in layman's language, usually our language, and they're easy to understand, okay? Easy to remember. They usually kind of describe the plant in a way that we can recognize. So we tend to like these. So for example, this one right here is called downy rattlesnake plantain. That's its common name. The other type of name is referred to as the scientific name. It'll be in Latin or Latinized. It's universal. You could use it anywhere in the world and people will know what you're talking about. It's the same name that they use in Japan, same name that they use over in China, all of it's the same. The formal, and that's because there's a formal system put in place for making these that everybody has to follow, no matter where you are or who you are. So the scientific name for this plant here is Goodyear pubescens. We'll get into what the rest of that part is in a little bit. Now, most students get sad when I tell them that the common names in some ways are kind of useless, because they like common names a lot. In many cases, what they'll do in the site recognition exam is they'll recognize the plant by the common name. So on the test, they'll say, okay, I know that this is downy rattlesnake plantain, and then later they'll link it up to the scientific name, which is perfectly fine. Eventually, you'll get to the point where you'll start just automatically thinking scientific name, but that's oftentimes the way it works off in the first place. But let's talk about why common names are actually a disadvantage. There's a lot of disadvantages to these. First names, they're governed by no rules. You can do this. You can go out in your backyard and name a plant. You don't even have to tell anybody what it is. Just name it, right? And nobody has to use that name if they don't want to. There's no rules. You can name it anything you want to. Feather duster, doesn't matter, right? Anything that you think it looks like. Absolutely no rules and nothing, and everything's fair game. Second, different cultures have different systems for naming plants. I'm using plants as an example here. This applies to any organism. So for example, some cultures base their names on how they use that organism. Right? If it's something that they use for food versus something used for housing, that may be how they name them. And then you run across one problem where, what if we don't ever use it? Oftentimes they don't name it. It doesn't have a name. We'll get into that in a second. There's also many, many different languages. Our common names are in our own language. As you saw, the Goodyear pubescens, the downy rattlesnake plantains, that's English. Downy rattlesnake plantain is English. But if you try and do that over in China, they're not gonna have any idea what you're talking about. They may not speak the language. And so it has no meaning to them. And so you have to make sure that you've got some sort of a formal system of everybody using the same language. We picked Latin because that was the language of the sciences, okay, back in the day. And it was kind of a neutral one that everybody could use. Nobody speaks it now, but that's what they decided to use for science types of terms. Here's another problem. Wide-ranging plants oftentimes have several different common names associated with them. So if you've ever seen this kind of a plant before, and this is just one species that's within a bigger group right here, most of us would call that a morning glory. Because here in the US, that's what we know them as morning glories. But if you go over to Europe and you start talking about morning glories, even in countries that speak English, they're gonna have no idea what you're talking about, because they don't call anything over there morning glories. Anything that's in this genus Ipomea to them is referred to as a woodbine, right? Everything. And so if they come over here and they start talking about woodbines, most of us are not gonna know what they're referring to either. If you say Ipomea, they will. Next, sometimes more than one plant can share the same common name. It can lead to a lot of confusions. Some of those confusions can be dangerous. I'll use this as an example. Hemlock. How many of us have heard of hemlock before? Right? What does it mean to you? What does it mean to you? Hey, cones on the eastern hemlock tree. How about for you? Poison. A poison. Excellent. Excellent. Two different ideas here, and you're both right. you're both not wrong, okay? There are two different types of plants that are called hemlock. You got your eastern hemlock tree right here that's called Tsuga canadensis. We'll learn that one actually next week. That's one of the ones we're gonna have to learn to sight recognize. It's got little tiny cones like this. They're extra cute. It's in the pine family, okay, Pinaceae family. Completely harmless. Not that I recommend you eat it, right? But it's harmless. Now this one over here, that's a different story. Conium, okay? Maculatum is something that's in the carrot family, the family Apiaceae. That is the poisonous plant that killed Socrates. And that is in fact a poison. Do not consume that one. And yes, we do have those around here. We also have water hemlock as well, which is also poisonous. Okay, so things, don't just go out eating things that look like carrots, because there's a lot of things in that family that actually are poisonous. But they both called hemlock. And so it may mean something different to somebody else. If you tell your neighbor, I'm gonna get you some hemlock, they might look at you like, I don't know about that. You're just trying to give them a tree, right? It just depends. So that can cause some confusion for that reason as well. The other thing to keep in mind is that common names don't tell us anything about the rank of that plant. We're talking about a genus, a species, okay? And nothing about the classification and how they're related to each other, right? So for example, St. John's wort, when you say that, some of you probably heard that for herbal medicines. People sometimes take it for depression, but it can refer to a whole genus of plants that are called hypericums, or it can refer to a specific hypericum, a specific species within that genus. So which one are you talking about? You're talking about the genus, you're talking about the species, okay? Here's another problem. We've got two here that have a common name, Rose of Sharon and Flower of an Hour. If I just heard those two common names, I wouldn't have any idea that these two plants are actually very closely related to each other. Not only are they in the same family, but they're actually in the same genus as well. They're both different types of hibiscus. Okay? Your scientific name is going to tell you that. I would know by looking at Hibiscus syricus and Hibiscus trinotum that they are related to each other. Rose of Sharon, Flower of Hour, wouldn't have any idea. So scientific names can be kind of useful for that reason. Now, many plants actually don't have common names. You'll go through in your book and you'll find a couple of those that don't have common names. Sometimes they've named just the genus but not the individual species. Usually that's done if something is just inconspicuous and rare. Very rarely do people run across it, so they just haven't bothered to give it a common name because nobody ever sees it. But they have been scientifically discovered and described in the scientific literature, so they do have a scientific name, even though they may not have a common name. Names can also be a bit misleading, right, in terms of information. So for example, this plant right here is referred to as Christmas cherry in terms of its common name. So you might think, yay, something I can make jam out of, I can go eat on some pies and things like that. Don't do it, right? This is in the nightshade family, which is notorious for having a lot of different species that are poisonous. This one, the fruits are toxic. They are not cherries. They're actually more closely related to tomatoes, but in this case, you can't eat them, right? So Solanum pseudocapsicum is not something that you want to eat and consume, despite the fact that the name has the word cherry in it. Right? Then we have poison oak, which can be very confusing because it's not even closely related to oaks. Oaks are in the genus Quercus. They're part of the beech family, right? This is not an oak. It's not a quercus, okay? It's gonna be a toxicodendron. And it's in the cashew family. It's more related to cashews than it is to actual true oaks. So again, the names can be a little bit misleading to us. You liking common names a little bit less now? You should like them, right? I like them too, don't worry. Okay, so what is nomenclature? This formal system that we're gonna have to use to name things so that everybody follows the same set of rules and has the same format to them. Well, nomenclature in general is the assignment of names utilizing some sort of system of rules. Specifically for plants, land plants, what this class is about, we use a system called the International Code of Nomenclature for Algae, Fungi, and Plants, the ICN. You know what the initials stand for. I know it's an awfully long name, and unfortunately they changed it just a few years back to make it longer to this. Okay? But it's important to know what it is, because if you look the name and you learn the name, you already automatically know what organisms apply to it. Okay? So where did this come from? These rules that we're going to go through one by one, okay, were developed and they've been revised periodically by a series of international botanical congresses. This is a group of botanists from different places around the world, different countries, that all come together every 6 years and meet and talk about things like this. Right? Are rules satisfactory? Do we need to change anything? Right? As an example, just recently back in 2011, they took away one of the criteria that's needed to actually publish a name. A very useful one that they got rid of too, because nobody wanted to do it. You had to come up with a description in Latin for your plant. Who could do that? Right? I'd have to sit there and I'd have to write the description out in English and then have somebody translate that for me. So they said, we're just gonna get rid of that. So they can revise things. But they are the ones that determine what the rules are and how those get adjusted accordingly over time, meaning every 6 years. They did not meet in 2023 because of the pandemic. They're actually meeting this next year instead. Now, here's something to really understand though, and a lot of people don't know this, but this set of rules that we use for naming plants, fungi, and algae does not apply to cultivated plants. Any plants that require basically human intervention, right, for their for sustaining populations is named by a completely different set of rules. Right? It's the International Code of Nomenclature for Cultivated Plants. That's why you get weird things and weird names of cultivated plants that have like X's in them and things like that. We're not going to see anything like that in lab, in our site recognition tests, because we aren't going to be dealing with cultivated plants. Right? And you're not going to use cultivated plants in your collections. So different set of rules. Okay, so we're going to go through the principles for the ICN one by one. And I'm never going to ask you to write out each of the statements that's a part of the set of rules, the ICN, because they're really long-winded and wordy and written by people who just like to listen to themselves talk. But I do want you to know what they mean. Okay? So what I'm going to do is I'm going to present to you what the wording is in the actual document first. And then I'm going to explain to you what it means. And that's what I want you to know. I want you to know the meaning, right? Not the actual sentence itself. The first statement in the set of rules in this document says that the botanical nomenclature is independent of zoological and bacteriological and viral nomenclature. This is one of the easiest ones to understand of all the different sets of rules. That means that this code that we use, this set of rules that we use to name things, only applies to organisms that in the past were traditionally treated as plants. That includes land plants, it includes algae and fungi. Yes, way back when, if you go back to some of your emeritus professors and ask them if they took a botany class, they're going to tell you they learned fungi in it. Because people say, well, they don't move. They're close enough. They're probably plants. We know that they're not plants, right? They're not even close to what plants are. They're heterotrophic, they're not autotrophic, right? So they're not even in the same kingdom. So nowadays, botany classes are not going to include fungi in it. But traditionally, they were treated as plants. And so we still use this set of rules to name them, along with algae, which are plants. We use this for both species that exist today, our modern-day species, as well as extinct species. So plants and things that we find in the fossil record, we still use this system for naming those. Same set of rules. Doesn't have to still be existing today for us to use. Okay, number 2. A botanical name is fixed to a taxon by a type. First time you read that, you kind of look at that like, what in the world are they talking about? Okay. First thing you're going to have to understand is what a type is. Okay? Anytime somebody names a new plant, okay, they had to have been looking at something originally, a plant that they found, to describe it, right? They have actual material that they've been studying for a long time. They've determined this is something that's new to scientific literature. It's a new plant. They've been looking at it, measuring it. They have a representative for this species, and they say, I think this is a new species. They're going to designate that individual, right, as a type, right? Which means it is now going to serve as the representative for that species. Anybody who wants to know which plant actually has all the perfect characteristics that describe this plant, right, will be able to actually go to and look at this original material. They'll actually be able to look at the plant. Okay? So they're designating a specific specimen as a type to serve as a permanent reference for a name. Permanent, meaning 50, 60, 100 years from now, somebody can still look at this plant. means it has to be preserved in the right way, in a way that's gonna stand the test of time and not degrade. That's where our herbariums come into play here. That's one of the reasons why they're so very important. Okay, so what sort of thing can be a type? Can they use as this representative of this species? Usually it's gonna be dried plant material that's been preserved and it's been archived in an herbarium. Okay? I will tell you that we don't have any of these because we're not big enough nor important enough to have them. Right? You go to Arwood institutions like Knoxville, they're gonna have some of these, and they guard them with their lives. They're very valuable because there's only one of them. Okay? We'll get into this. So that means that this, for example, this is gonna be an isotype, we'll talk about that a little bit later. Okay, that's gonna be a unique specimen that is gonna be something that's representative for the species. Okay, it's been preserved in your herbarium. They used to also allow illustrations or preserved cultures. So not necessarily something that's dried and pressed like this, it could be in a jar of formaldehyde or something like that. I don't really use formaldehyde before, but they did back then. That could also be considered a type. Typically now at this point, kind of from like 1958 on, they kind of prohibited this. Right? Very rarely do they allow illustrations or things like this to serve as types. They're gonna want something like a press specimen that's in the herbarium. Now there's different types of types. Try saying that 10 times. Types of types. Okay. There's a holotype, probably the most important. Isotype. There's electrotype and a neotype. There's actually more than that. Those are the only four we're going to talk about. Okay. Holotype, isotype, electrotype, neotype. You may have heard of these before? Perfect. All right, let's start with the holotype first. Is your question?
Well, I was just gonna ask, like, if plants were discovered, like, per se, in a fossil, if it's like in a rock or something, then how would they preserve that in a way? Like, would they just
In the
take
fossil,
that?
yes, in the case of the fossil, you're gonna have to have the preserved fossil itself.
Gotcha.
That's gonna fall in that category. Very good question. Technically, that's still the plant, the carbon's still there, it's just no longer in the same form that it was when it was alive. Or maybe impressions too. That's a good question. All right, so let's talk about our holotype first, and why this one's so important. Right? There's only ever gonna be a single holotype, one single plant specimen out there. That would be the holotype. This is the type, okay? This is the one that the person who published the name said, this is the most perfect representation of this species that I can find. This is what I was originally looking at when I named it. These are the characteristics that I described. This is what you wanna come to and reference if you ever wanna know what the perfect specimen looks like. So it's the single specimen or illustration designated as the type of the species, the representative by the original author at the time the species name and description was published. They're very valuable. And nowadays, when you publish a name, you've got to put this in there, in that publication. You have to tell where that holotype is located and designated, what herbarium it's in, geographic location, all of that information, so people can actually go to that and see it. There's only ever one of these. But sometimes you want some backup, you want some reassurances, because if you put this in one building and that building burns down, that's a problem, right? And that does happen. Herbariums do fall down. Everything's made of paper and dried plant material, so it's gonna go up pretty fast. Right? But if you have some copies, that would be useful. They cannot be considered the holotype though. Those are designated as an isotype. Oftentimes the person who's publishing this has gone out and collected a group of plants all at the same time by the same person in the same population. Has to be the same time and place or it doesn't count as an isotype. They've collected copies. They've picked one of them to be the holotype. All the rest of them are isotypes, and they generally do not house them in the same herbarium that the holotype is in. They spread them out. So in case something happens to the holotype, they can kind of dig into this other material. All right, we'll stop with this one. We'll get into the other two after this. Just keep that isotype in mind because that's going to help us figure out what the other two types are in the next class. You have a quiz coming up soon, it's listed for Wednesday next week, so we have a lecture before that time. I just wanna put that on your radar. We're not as far behind as we could be because we moved through the syllabus on the first day quickly.